Yes, sorry. So first I have to apologize for the change of topic. I was supposed to present this nice work by Luli and with the assistance of Melanie Stephan that was published last week in PLOS ONE. But it deals with calcium and calcineurin and CAM-KINase2, and you already heard a bit about that. Melanie is with us today, so if you want to talk about this work, which is really great, that shows plenty of interesting things like both calcineurin and CAM-KINase2 are actually increasing together rather than a balance, and it's a ratio that changes. That you have, because of the allosteric property of calmodulin, you have stabilization, you have a response that outlasts after a certain frequency much the calcium signal and so on. So please go and talk to Melanie. But instead of that, I will effectively talk about automatic generation of models. And the reason is the following. So at the moment, we have, and that will be the topic of this afternoon's workshop, I guess, you have a huge amount of information about pathways, networks, and so on in the literature. And you have also a lot of information stored in databases all over the world, curated or non-curated, many of them specific to neuroscience, but not only. However, in the immense majority of the cases, the modeling process is that one scientist picks a few papers out of the literature, write down a model either using processed representation or directly equations, and does its own modeling and simulation exercise. And the question is, what about all this information here and all the information in the database? That is basically there, but we don't use it. We restart from scratch all the time. And I think that's very valid. I mean, there is a lot of interesting modeling and simulation to be done that way. But as we gather more and more data, we need to have other methods to generate models. So from structural genomics, interactomes, and as Mary already showed, another version of this one from knowledge on pathways. Also, we now are able to build much bigger models. So you may have seen this paper in July in Cell on a model of mycoplasma. This is not neuroinformatics, but that's a game-changing paper where people try to model a whole cell, really a whole cell, all the genes, all the molecule, and after the movie of Thomas, even if that was not the right one, I mean, I think we're maybe not far from that. And there are examples as well in the literature where large curated pathways gave rise to many different models where people started from an existing pathway. So that's the curated version of the the yeast metabolic networks published a few years ago. We have now submitted the human one. And for the last 4 years, plenty of papers were based on this, where people pick part of the network and build models. So because I will mention models and pathways a lot, I need to define what those are. And I will base myself on Wikipedia. As we saw yesterday, Wikipedia is a good source of information, a reliable one. So Wikipedia tells us that in biochemistry, metabolic pathways are series of chemical reactions occurring within a cell. Okay, and that will be our operational definition. You have plenty of pathways databases, but the important thing is that those pathways are detailed representation of reality based on observation. They are meant to represent the reality, okay? But when you do modeling, this is not always what you want. And Wikipedia tells us that a model is just a description of a system using mathematical concepts and languages. So you have variables, functions, constraints to limit this model. And the important thing is that this model is an abstract representation of reality, right? Based on needs, you develop your model based on the question you ask and the data you have to generate the model and validate the model, and that was already mentioned this morning. So the whole project, the idea was to start from pathways, possibly in proprietary format, transform them in standard format, either to represent the models or for a computer or for a human being, generate mathematical models and put everything in a database for everyone to be able to use it. And I will, so a bit of SBML, and already some people in the room maybe say, oh no, he will talk about SBML again. That will be just a few slides. So SBML, the same biology markup language, is just an XML language to encode models, right? And then you can exchange them. It's very simple. You basically define your variables, the compartments, the molecular species you put in there, the parameters, and all the relationships between those variables. You can represent various containers, pools that react within containers, between containers. You can put discrete event, arbitrary rule, and so on and so forth. So that's a micro SBML model with two species A and B, right? In this, a compartment cell, one parameter, and then I can describe a mathematical reaction, a kinetic law here, regulating the velocity of the reaction. And if I load that in various software, for instance, Copazi or Cell Designer, and I click run, I get the same result. And that was not possible before SBML. So SBML now is the level 3, and the important thing is that in level 3, you have a modular language with various packages covering different aspects of models or different type of model. And today we will talk about the core package, which is shared between everyone, the layout package, and package to encode qualitative models, logical models, Petri net, and also flux balance constraints. The other standard we will use is SBGN, the Systems Biology Graphical Notation, which is a set of 3 languages to represent biological pathways. And I won't deal in the difference between those 3 languages, but you will see why we need them. And I will present an example based on KEGG. So we did actually the same workflow on other database. We just published the NCI PID database to model transformation. But KEGG was important. So the Kyoto Encyclopedia of Gene and Genomes has a subpart called KEGG Pathways. If you organize a course in systems biology, the first thing the student want to do is to go to KEGG, because it's nice graphically, and download KEGG information. And the second thing they do is to convert KEGG information from this proprietary format that cannot be used anywhere into SBML. So that's why it's an illuminating example. So you have 2 types of maps in KEGG. If you take a metabolic network, for instance, the TCA cycle, that is very, very important for the brain. Remember, brain, 2% of your mass, 10% of your energy consumption, I think. And so in those maps, you have those bubbles here that represent chemicals. And those chemicals are transformed into other chemicals using reactions. And this annotation is an enzyme classification number, right? So, and we have another way to represent pathways in KEGG. If you look at the signaling pathway, you have a complete different representation where you have those rectangles here, annotated with something that vaguely look like a protein name, and those have arrows connecting them together. So this is a bipartite graph, right? You have 2 types of nodes. This is a graph with only 1 type of node. This is a positive regulation. That will be a negative regulation, okay? So it's important to understand the difference between those two graphical representations. It's a fundamental piece of information to understand systems biology. The first representation is a process description. It's a classical biochemical representation used for the last 70 years or so, where you have 2 pools of molecular species that are transformed into other pools through processes, and those processes can be regulated by yet other pools, okay? Bipartite graph. This node never connects to this one never connects to this one. So those representations are directional. They are sequential. You can start from somewhere and somewhere else. They are mechanistic. You know exactly what's happening. But they are subjected to combinatorial explosion, something alluded to by Thomas. They're used a lot in process modeling, for biochemistry, and so KEGG metabolic pathways are in process description, or Reactome for those of you knowing this database, and we will encode that in SBML core. The second type of diagram is an activity flow. So this is directional as well, sequential as well, but it's non-mechanistic. When you have that, you don't know what RAF does to MEC. RAF stabilize an active state of MEC, stimulate the expression of MEC, inhibits the degradation of MEC, you don't know, right? So you just know that RAF, so this is actually an activity. What you represent here is RAF activity stimulate MEC activity in some way. It can be used to build logical models, and it's used a lot for signaling pathways, KEGG signaling pathways, but also STKE, for instance, use the activity flows. And we'll encode that in SBML-Qual, the package used to encode logical models. This is a complete workflow. It's a bit more complex than the previous one. So we'll have 3 different sub-workflows. The first one is to take all KEGG metabolic networks, convert them into SBML core, represent them graphically with the SBML layout package, and converting them into SBGN for the graphical representation, transform those into chemical kinetics models, put them in a database. A second workflow will take the non-metabolic pathway, the signaling pathways basically mostly, enrich them with information coming from other databases. We use BioCarta in this example. Generate SBML-Qual, same, we generate the graphical representation with system biology graphical notation activity flow, and generate logical models, distribute them. And finally, we'll have a third branch when we will take the metabolic networks in KEGG for a given species, reconstruct whole genome metabolic networks, enrich them with information coming from Metasite, which is another database of metabolic reactions, and generate models called flux balance constraints model and distribute them, okay? I will start with the whole genome reconstruction. So we take the information from KEGG, we extract it, we convert it into SBML, and we annotate them with so-called identifiers.org URI, basically URIs that identify exactly all the elements of the model. We do the same for Metasite. And then we will go through the workflow with those SBML files and the identifiers that will be our handle, that will be able to relate information. So we merge the information from KEGG, we protonate on both sides, we mass balance the equation, and then we merge them, the information coming from KEGG and Metasite, thanks to the identifiers. We add the transporters. You will see in a minute why it's important. We add the biomass, and then we generate the FBA models. So what are flux balance constraint models? The idea is that for any metabolic network, you can represent a matrix, which is a stoichiometric matrix, okay? You have on one dimension all the metabolites, on the other dimension all the reactions. So this is a small metabolic network. We have our small matrix. -1 means it's consumed, 1 is produced, and 0 means this metabolite is not involved in this reaction. So the first thing for FBA is we need to add the boundary condition, okay? The input and output, so that inflates a bit our matrix. Once we have that, we'll go further. So for each reaction, we can have a velocity, okay? And so we have a vector of velocities that we will work with. We don't need the kinetic rate law for FBA. The only thing you need to know is there is a velocity, a number. And then you do a steady state analysis. So the steady state analysis is basically the multiplication of the stoichiometric matrix by the velocity vector equals 0. That gives you all the possible steady state of your network. But that's not enough, because generally our model is underdetermined. So you can't determine all the velocities just with your information. So you need to add objective function. For instance, you will say, I want to maximize the production of ATP. Or if you are brewing beer and you're using Saccharomyces, you say, I want to optimize the production of ethanol. And then you can solve that. So that will be the result of the steady state analysis for those 3 velocities. If you put your objective function, you can reach a given vector, okay? So now for individual pathway, I said we will generate chemical kinetics model. So we do that with a workflow based on something called KEGG translators. So we generate SBML core, we clean the reaction using the KEGG API that allow us to interrogate all the different databases of the KEGG constellations. And then we will do 2 things. We will interrogate CBORK, that is a database of chemical reactions. If we have mechanistic rate law for this reaction, we reuse it. This is unfortunately not the case for the majority. So for the majority of the case, we will use modular rate law. So modular rate law is something that's been presented by Wolfram Liebermeister in the group of EDACLIP in 2010, and this is great. This is the absolute general rate law that represents any possible rate law for a chemical reaction. And then you have variants. For each of those bits, you have variants. And so, for instance, here, this will be a generalized representation of mass action law. And based on the data you have to decide the granularity and the generality of your rate law. Okay? And for the signaling pathways, remember, we don't have metabolic networks. We just have activity influencing other activities. We'll use logical models. So logical models, briefly, you have different activities here, A, B, C, and you put logical rules, right? If A and B, then C. If C, not B, and so on and so forth. So you end up with vectors of activity. Here I use a Boolean, but some people use multi-valued models as well. So if you have 1, 1, 1, then B is inhibited by C, C is 1, then B becomes 0, and so on. And you can generate a state diagram and identify the various behavior of your network. Right. So we released the first version of the models in May with the 22nd release of Biomodels database. We have, so big numbers. We have plenty of models now. We have about 150,000 models that represent, I think, a bit more than 10 million reactions. And importantly, they are annotated with 400 million cross-references that allows you to identify specifically which metabolite is which, and so on and so forth. And if you go to Biomodels database, there is now a new branch called Path2Models, and you can explore them, browse them, or explore them with taxonomy as well. That's an on-try in the database for the glutamatergic synapse. You even have the representation coming from KEGG. And I would like to end up here with acknowledgement. Those three chaps have generated most of the data. So Neil was in charge of the metabolic reconstruction, the whole genome metabolic reconstruction. Clemens of the chemical kinetics model. Finia of the logical models. Tobias took care of the graphical representation of the process-based models, Michael of the logical models. Andreas was the PI on the Tübingen side. Camille is the coordinator of Biomodels database and all associated activities. But actually, we had many more people involved in this work, because that was a pretty large work. I won't enumerate all of them, but they will be in my presentation. And I would like to end with an advertisement for a book. So for the few of you who are not authors of this book, all the others are probably in the room. So systems biology applied to neuronal system is a very wide topic. I believe in this workshop we will address mostly biochemical systems, but we have many more systems at different levels. So there you will find information from transcriptomics and proteomics all the way to very large network and modeling of EEG and so on and so forth via classical spiking neurons or multi-compartment neurons, etc. Thank you.